Hello again, everyone. Welcome back from the break. What I'd like to do now is move on to our final session of the workshop. We're going to split this into two different topics, actually, just to try and cover as much as we can in the duration of the course. So, the first part of our session is going to be looking at the BioMart data mining tool, which we can access from the Ensembl Genome browser and use to export tables of Ensembl data. So what I want to do in this presentation is explain to you about the BioMart tool, explain what it is and what you can use it for. And then we're going to move on to looking at how we can actually use the BioMart interface. So what we have, what we call the BioMart principle, which is makes it sound a bit fancy and complicated, but really it's just 4 simple steps that we need to execute to be able to submit our query and get our table returned to us with the data. And then we'll move on to having a demo that we'll all do together, and then there'll be a chance for exercises at the end as well. So BioMart is a tool in your browser that allows you to export Ensembl data, but without any programming required. So as you know, I said at the beginning of the course, there are more advanced data access methods using the Ensembl Perl and REST APIs that require programming knowledge in either Perl or another language such as Python or R. But BioMart is a tool that has a web interface. So it allows us just to build queries with a few simple mouse clicks. And then at the end, it can generate a customizable data table for us to export, which we can then obviously continue working with outside of Ensembl. So BioMart is very useful. BioMart's a very useful tool, and I like to tell people that it's a great time-saving tool because what I tend to think of it as being useful for is for doing things that would be really time-consuming, difficult, or boring in the Ensembl Genome browser. So if we could think of an example where we have perhaps a list of 100 genes that we're interested in, maybe we've done an experiment looking at differential expression, and we find that 100 different genes are differentially expressed in our experimental conditions. We could go to the Ensembl browser and look at the gene tab and retrieve information for those 100 genes one by one, but that's going to be really slow and laborious. So what we can actually do is use BioMart to retrieve information about that set of genes. So we would simply give BioMart our list of genes and get a whole wide array of different types of information about those genes in our data table. So the idea is that we can query multiple things at once. So those things might be a set of genes, like in the example I just gave, or it might be a set of variants, or it might be a set of regulatory features. And then out of the other end, you can get IDs from different databases. You can get gene locations, and you can get sequences as well, for example. This is just some of the main different types of things that you can get with BioMart as a data output. But in the demo, I'm going to show you as many different types of things that you can retrieve from the BioMart tool. And the idea is that you can obviously begin to export large amounts of data. But this final point has an important caveat, and it's that BioMart is really designed to be used for medium-sized data sets. So anything up to around about 500 genes or variants or regulatory features at a time is sort of our limit that we suggest. If you're trying to perform, if you're trying to retrieve data for all of the human genes, for example, then that won't work, and the BioMart query will take too long and will time out. So if you do want to do a genome-wide query, there are other methods that you can think about that I'll touch upon at the end. But this is really for medium-sized queries. So if you want to use BioMart, you have you can use it through the Genome browser. So you can find the link for the BioMart tool in the blue bar at the top of the homepage. You can see I've just highlighted it here. You probably would have maybe noticed it when we were going through our demos on the previous sessions as well. We also have BioMart for the Ensembl genome species as well. So this is an example of the BioMart link in the header for the Ensembl metazoa pages. And we also have BioMart available for plants, fungi, and protists as well. So you can see Ensembl BioMart for the vertebrates, plants, fungi, metazoa, and protists. The only group of species in Ensembl that we don't have BioMart available for is the bacteria. So the reason for this is that we have so many bacterial genomes, many thousands of bacterial genomes available through Ensembl Genomes, that really the BioMart databases and interface can't deal with such a large amount of data to search. So in the future, we do hope to extend this BioMart functionality to bacterial species as well. But at the moment, we're just thinking about these vertebrates and then these specific non-vertebrate species as well. So if you want to use BioMart, as I said, we have this 4-step principle that we can always follow, and it helps us to build our query and make sure that we retrieve the data that we want. So step 1 is to choose your data set and database. Step 2 is to use filters to narrow down the data set. Step 3 is to choose the attributes, so pick what you want to have returned to you about your features of interest in your data table. And then step 4 is to view the table and download the results. So what I want to do now is just go over these 4 steps in a bit more detail to explain how each of them work for you. So step 1 is the data set. So firstly, you define the type of database that you want to search. So that might be genes, variation, or regulation, most commonly. And then you also need to choose your species of interest. So in step 1, you would say, for example, I would like to query the human genes database, as an example. Step 2 is that you then use filters to narrow down that data set to specifically choose the features that you're interested in. So in step 1, you say, I'm interested in the whole set of human genes. And then in step 2, you say, I only want to retrieve information about this set of human genes. So within this full data set, I'm filtering it down, narrowing it down to perhaps a smaller set of a smaller subset of genes. So we can use filters to narrow down that data set with a number of combinations of parameters. So your filter might be a genomic region, or it might be a list of IDs or gene names or symbols. It might be a particular biological function. So you might use a Go term as a filter to retrieve information about genes that are associated with a particular Go term. Or you might already want to use a phenotype as a filter, so to retrieve information about genes with a specific phenotype. Now, importantly to remember is that you can build these queries to become more and more complicated. So you can use more than one filter at a time, but you have to remember that you're only going to retrieve information for features that satisfy all the filters. So for example, this is a simple query with 2 different filters. If I said in step 1, I'm interested in the mouse genes, if I then said filter 1 is perhaps a genomic region, chromosome 9, the whole of chromosome 9 as my first filter, and then filter 2 might be with a transmembrane domain. What I'm actually going to retrieve is information for mouse genes on chromosome 9 with a transmembrane domain. So I'm basically going to retrieve things that satisfy all the filters at the same time. So I'm only going to retrieve attributes for those genes. Step 3 is to then choose your attributes. So this is where you define the data that you want to retrieve for the things that you defined with your filters. So you say, of the things that I chose with my filters, this is what I want to retrieve about them. So you might want to get IDs, so from Ensembl or from external databases. You might want to get information about the gene features itself, like the exons and the transcripts. You might want to get the sequence of the genes and the transcripts, for example. And you can also get ortholog and paralog predictions as well. Again, this is just a summary snapshot of some of the things that you can retrieve, but we'll go through this in a bit more detail in the demo as well. And then finally, the final step is to view the results. So you can view the results in the web interface, and then you can download them in HTML format, TSV or CSV file formats. Excel is also quite a common file format that people like to download data tables in as well. And then FASTA for sequences. Obviously, what this means is that you can then export the table into your own software, and you can begin filtering, combining it with other data sets, analyzing the data outside of Ensembl. So what we're going to use in the demo is the BioMart Web Interface, but there's also an R package that you can use to query the BioMart databases. So we won't look at this in the demo today, but if you are familiar with using the R programming language, you can install the Bioconductor BioMart package using the links and the documentation on this page, and perform BioMart queries just like we're going to do in the demo, but using R. So what we're going to do in our demo is have a look at a set of 6 human genes, and we're going to find out their NCBI IDs. We're going to find out their function by retrieving the Go terms that they're associated with, and we're going to get their cDNA sequences as well. So just to quickly go through what these 4 steps are going to be for our query. So step 1 is the data set. So we know that we're interested in human genes, so that will be our data set, the gene set, and human Homo sapiens as our species of interest. Then the filters are actually going to be the gene names themselves. So in fact, we're going to have a really simple query just using a single filter. And so what we can do is, what we're essentially saying is, of all the human genes, I'm only interested in the ones with these names. Step 3 is to then choose our attributes. So these are the things that we want to retrieve: the NCBI IDs, the Go terms, and the cDNA sequences. And then finally, we're going to get our results as a data table. So if you want to follow along with me, the screenshots start on page 51 of the course book. Sorry, if you want to follow along in the course book, that's where the screenshots start on page 51, but you can follow along with me as well. If you do wish to follow along with me, what we're going to have to do is just get our list of genes that we can copy and paste into BioMart. So what I'm going to do is navigate to the course materials page and just click on the copy sheet file that we used yesterday. So this is the file that we used for getting the VEP data for the demo yesterday, for example. But you can see here as well, there's a section called BioMart Demo. So I'm just going to copy this list of genes to paste in at the right point in my demo. So hopefully, if you want to follow along, you will have that. And then we can navigate to Ensembl. And then I'm going to click on BioMart in the blue bar at the top to navigate to the BioMart tool. And this is the interface, and it will guide us through these 4 steps. So we can see that there's not much on the page to start with. We just have this single drop-down box, and it's asking us to choose our database. So at the moment, we have Ensembl Genes 102. I think that just based on the chat box, there were also people who might be interested in retrieving information about regulatory features. So in that case, you would pick Ensembl regulation to get information about the regulatory features. But we're obviously interested in genes. So it's really important to think about which database you select, depending on what type of features that you want to get information about. So we'll click genes. Then the next drop-down menu is asking us to choose our data set, which is our species. So we can click there and select human from the drop-down menu. So that's now step 1 completed. What we can do now is move on to step 2. And to do that, you can see the menu on the left-hand side has now begun to be populated with our options for selecting our filters and our attributes. So the first thing we want to do is to move on to step 2, which is the filters. So we can click on filters. So when you click on filters, we can see all of these expandable menus now appear in the main part of the page, which allow us to view and select the filters that we need. So I won't be able to go through them all, but I just want to open a few of these menus just to show you the types of filters that you can use. So if you open the region panel, you can see that you can filter by chromosome number. So if you want to filter by chromosome 9, for example, all you would do is click on the box and select the chromosome number. So as you select the filters, you can see that they're added to the filter menu on the left-hand side. Now, this is not what we want to do. So what you would have to ensure is that you then click on the button to remove this filter again. So as you begin playing with BioMart and clicking on filters and seeing what they do, you need to make sure that if you turn them on and then you don't want them, you need to turn them off again. So I don't want to use the chromosome number as a filter. I just wanted to show it to you as an example. Beneath, you can see that you can also filter by specific genomic coordinates. You could filter by a specific karyotypic band, genetic markers, or you can filter by multiple genomic regions as well. If we open the genes panel, so this is the next menu underneath, we can see some of the filters that are associated with genes. So this is actually where we want to find our filter of interest. So this is our filter that we're going to use, the input external references ID list. So we click on this box to select it as a filter. Then we can paste our gene names into the text box. And you can use a number of different types of separator here. So commas, carriage returns, and that would work as to make a list of genes. And then the final thing that you have to do is to ensure that you tell BioMart what format your list of IDs is in. So we have gene names. So what we need to do is click the drop-down menu and select gene names from the list. Now, before I do, I just wanted to sort of use this opportunity to show you the wide array of different IDs that you can use as a filter here. So if you have IDs from any of these different databases in any of these formats, you can actually use them as a filter to retrieve information. So this just really tries to show you the versatility of BioMart as a tool. So if you've got IDs in a number of different formats, then you can use them as a filter. But we have the gene names. So we can click on gene names here. And now that's step 2 actually done. Before we move on to step 3, I'll just show you some of the other filters. So you can filter by gene type. So protein coding genes, for example, you can select if you only want to see information about protein coding genes. You can get information about genes that are part of the main select annotation, or with a main select transcript, sorry. So Isabel is just asking, are these gene names? I thought they were gene symbols. So yes, the answer is that names and symbols are synonymous with one another. So these are gene names. For human, we equally could have picked HGNC symbols. So we could have picked this because this is the Hugo Gene Nomenclature Committee, so the committee that's responsible for naming human genes. So in this case, we could have picked gene names or HGNC symbols and it would have worked. If we were looking at another species that gets its names not from HGNC, then we would have we would need to click gene names. So hopefully that answers your question. Okay. As I said, if we scroll further down, you can also filter by phenotype. So you can pick a phenotype that you're interested in to get information about genes associated with your phenotype. And you can also open gene ontology and filter based on Go terms that you might be interested in. So you can use these specific Go accession numbers and use those as a filter. Good. Okay, so obviously there's lots of filters here and we haven't gone through them all. So in the exercises and in your own time, what you'll have to do is have a look through the filters and think about how you can use them, use the different filters, use them in combination with one another to really create the data set that you're interested in retrieving information for. The next thing to do is to click count. So that's the the blue button in the middle at the top left-hand corner. And if you click count, this will just allow you to allow you to check that your filters have worked correctly. So what we can see here is that we're looking at 6 out of 67,139 genes. So what BioMart's telling us is that there are 67,139 human genes, and we've narrowed that data set down to just 6. And that we obviously had a list of 6 genes, so this makes sense for us. If this number was 0 or 67,139, then we'd obviously have some questions because it suggests that BioMart's not reading our filters correctly or in the way that we intended. So that's now step 2 done. So now we can move on to step 3 to select the attributes. And when we click on attributes, one of the most important things to notice to start with is that the attributes are divided into these 6 categories that you can select with this radio button option here at the top. Now, importantly, you can only select attributes from one of these categories at a time. So what this might mean is that, well, what this is going to mean for us, in fact, is that we have to perform 2 BioMart queries, one after the other, to firstly get information from this features section, and then we're going to have to come back to the sequences to get our cDNA sequences. But I'll show you how you can fast track this, do a second query quickly afterwards. So let's stay in features for the time being. So if we open gene, we can see that some of the attributes have been selected by default. So we have the gene stable ID and gene stable ID version, and the transcript stable ID and the transcript stable ID version already selected. I'm also going to select gene name because that will allow me to see in my output, it will allow me to see the names of the genes that I used as my input, so I can match up the rows to the names of the genes that I used in my input. And then in external, you can see that we have a section about Go terms. So this was one of the things we wanted to retrieve. So we're going to get the Go term accession, name, and definition. And then if we scroll even further down, we can see that we have external references. So we can get IDs from a number of different external databases. So I want to get the NCBI ID. Where would that be? Okay, here, yeah. So we want the NCBI gene ID. So obviously, you can see that there are lots of different databases again. So you can pick which ones you want to get the IDs from. What you'll have noticed is that as I've been clicking on the attributes, they've been added to the list of attributes on the left-hand side here. So now you can see a list of all the attributes that are going to be in our table. So we're essentially just going to have our table with a column with information about each of these different attributes for each gene. What you'll have noticed as well is that the order that I clicked them is the order they appear in the list, and the order that they appear in the list is the order that they're going to be in the columns of our table. So if you have a particular downstream use case in mind where the order of the columns is going to be important, then this is going to be the right time to change that. So you could, for example, remove the Go IDs and put them back in. And now you can see that the NCBI gene ID is moved up the list. So now that column in our table will be before these Go terms. So the order that you click them is important at this stage. So that's now step 3 done. The final thing to do is step 4, which is to click the results button in the top left-hand corner. And here we can now see a preview of our results table. So we can see we have a view of just the first 10 rows. And this is our data table. So we can see these are our attributes: gene stable ID, gene stable ID version, transcript stable ID, transcript stable ID version, and then the gene name, the NCBI ID, and then the 3 different Go attributes as well. Now, if you want to see this full table, you can click on the drop-down menu here and click All. And that will take you to a new tab. So it will open up a new tab in your browser, and it will load the full data table in the web for you. So you can actually begin to view the full table. Now, importantly, what we're going to see is that even though we started with just a list of 6 genes, even though we started with just a list of 6 genes, we now have a table with many, perhaps hundreds of rows. And that is because each row of this table is unique. So each of our 6 genes is going to have multiple transcripts, and each of those transcripts will be associated with multiple Go terms. So what you can see here is that this row of the table is referring to this specific transcript of BRCA2, which is linked to this specific Go term. This row of the table is referring to this transcript of BRCA2, which is linked to another Go term. So what we can actually see is that we have many rows of our table. The first set you can see are all linked to transcripts of BRCA2, but if we scroll down far enough, we will eventually see that we're now looking at transcripts of MH9, and even further down we'll be seeing the other gene CIST2, for example. Okay, so if we go back to the main BioMart web page where we started, what you can begin to do now is downloading the table. So obviously we just saw that the table is really large, and so exporting it for you to further filter and manipulate it is going to be important. So you can use the option at the top here to download the table. So you can choose to download it as a file, a compressed file, or a compressed web file by email. So I would always advise you to download it by email, because it means that once you choose to download and press go, it will package the file up, the data table up at our end in the Ensembl servers, and then send it directly to your email just a few minutes later. So it means that there doesn't have to be this continual connection between the Ensembl servers and your own computer as well. So I would always choose by email. Then we choose our file format, so TSV, CSV, HTML, or Excel, for example. And then we would just put in our email address, for example. And then we would click go. So this would then email a table of the results directly to us. The final thing to do, as we looked at in the presentation, was the sequences. So that was when we were looking at the attributes, we knew that we had to come back and get the sequences in a second query. So what we can actually do is just click back onto attributes. So we know that step 1 and step 2 have been completed correctly. So all we have to do is click back onto attributes, go back to the top, and we can begin selecting our new set of attributes to reperform the query again, getting new data. So now we can click on sequences, and then in the expandable menus, we can open the sequences menu, and now you can see the different types of sequences that are available. So by default, we're going to get the peptide sequence, which isn't right for us. We want the cDNA sequence, so we can click there. And you can see just highlighted on the diagram, just to try and explain to you the sequence we're going to get. So we're going to get the sequences of all of the exons, coding and non-coding, so the cDNA sequence. Also, if we open the header panel, you can also see that in the header we're going to get the gene stable ID and the gene stable ID version. And for this example, I'll also add the gene name to the header, because then I will be able to see what name of the gene is being referred to with each of the sequences. And then finally, I can click on results. Just go back to step 4 again to get the results. And now you can see these are the cDNA sequences of all the transcripts of all of my genes. So at the top, I have the cDNA sequence of one of the transcripts of ESPN, and then the cDNA sequence of one of the transcripts of THRB, one of the other transcripts of THRB, going down and down. So again, I can either view them in the web, or I can download them, but this time only as FASTA, because this is obviously sequence data. FASTA is the only format that we support for downloading sequence in BioMart. To do the exercises, you can go to page 15 of the question book. So just find it again. Here we go. So we're on the question book now. So the pages are on 15 and 16. So we have 1, 2, 3, 4, 5 different BioMart exercises. And if you look in the question in the copy sheet as well, you can actually see that we have some of these IDs listed out for you so that you can copy and paste them directly from the copy sheet as well. We're going to be looking at how we can visualize our own data sets within the Ensembl Genome browser. So it's important to remember in this module that what we are not doing is submitting data to Ensembl as a way that it then gets integrated in the Ensembl database and everyone around the world can see it and view it. This is a way of visualizing our own data sets against the publicly, alongside the publicly available Ensembl data. So if you want to view your own data in Ensembl, you can do this. I've already alluded to it in the session on Tuesday, but you can use this blue custom tracks button in the location tab to upload your own custom track. So we support a number of different file formats. So this is just an example of some of the more common file formats that we support. So if you've got some alignments in BAM format, for example, you can upload BAM files. You can upload gene features in GTF or GFF file format. You can discrete genomic regions in BED or BED graph files. Variants in VCF, for example, as well. So I do know that someone, I'm not sure who it was actually now, but someone asked about the file formats further back in the course during the BioMart session. And Emily actually, here we go. Emily actually provided a link of the different file formats that we support and you can use in Ensembl. So I'll put that link again back in the chat box so that, I'll put that link back in the chat box. It's not copying for me. What I'll do is I'll navigate to the documentation and find it during our during the demo, and I'll paste it into the chat box for you then. So we have our we have our custom tracks button that allows us to upload the data. So attaching your data, you can either upload it directly. So if you've got a file on your machine, you can upload it to Ensembl in the web interface. And the data is actually saved locally by Ensembl. And there's quite a small file size limit, so 20 megabytes limit. So it's not possible for large file formats. So for example, if you have BAM alignments across a whole chromosome, then that won't work for using the direct data upload. However, you can attach a remote file using a URL-based link. So if you have a public directory, such as an HTTP or an FTP public directory, you can attach a remote file that's stored there and there will be no size limit. So in fact, actually, the link that I was trying to copy and paste is directly here for you. So I won't actually need to do that. So you can use the link here. So if you do want to host URLs for larger files, if it's bigger than 20 megabytes, for example, then you need to attach it remotely. So you can do this if your institute has an FTP server that they can store the files on. You can use your own institute's FTP site, or you can use Cyverse or Figshare. So some people sometimes ask us about Google Drive and GitHub, which, well, Google Drive won't work. GitHub has not really worked in a few different people's hands. So these are the main solutions that we suggest to use if you want to create a URL with a large file that you want to attach remotely. So in our demo, what we're going to do is we're just going to visualize a small bed file by uploading it to the Ensembl Genome browser. So what we have in our bed file are 3 discrete genomic regions that map to large-scale deletions from patients that have presented with microcephaly and developmental delay. So if you want to follow along in the course book, the screenshots start on page 56, but you can also follow along with me as well. If you want to follow along with me, we'll have to open up the copy sheet again just to get the bed file that we want to copy and paste into the browser. So we can click on copy sheet here, And if we scroll to the very, very bottom, you can see this section called Add Your Data Demo. So you just need to copy these 3 rows of data. This is data in the bed file format, which we will look at in a moment. But for now, you can just copy these and we'll be pasting them at the right point in the demo. So to add them to Ensembl, we just go to www.ensembl.org, the main genome browser we've used before. And what I think I'll do is, I will do this for you now. We can click on Help and Docs. I'm just trying to show you how you can find information about the supported file format. So this is the Help and Documentation section. If you click on Help and Docs in the blue bar at the top, it will take you to our documentation section, which I'm going to show you again later as well. And then on the left-hand side, there's a lot of information. But if you look at the top, you can find this section saying adding custom tracks. So this shows you how you can, well, there's an actual tutorial video that shows you how you can do it as well. But then further down, there's information about all of the supported file formats. And if you click on them, there's actually more information about the specific formatting within each of those files that you need to have in order to upload to Ensembl. So here we go. This will go in the chat box now. Good. Okay. So if you want to upload your own data, as I said, you need to use the custom tracks button in the location tab. So what you can do, you've got a number of options. So if you know that you want to upload some data to human, you can select human from your species list. So you can either click it within the list here, or you can go to view full list of all species like we did on the first demo on Tuesday. Then search for my species of interest. To navigate to the species-specific homepage. So when you're in your species-specific homepage, you can click on the link here in the box in the top left-hand corner that says display your data in Ensembl. So you can either click this, which I'll do now, which will allow you to upload your data in the pop-up window here. Or if you are viewing a location within Ensembl, so if you're viewing an example location, we'll use this one here as a demonstration. You can use this custom tracks button when you're in the location tab. So the 2 ways that you can find the button, firstly from the species homepage for the species that you want to upload data for, or you can navigate to the location tab anywhere in the genome of the species that you're interested in and click on the custom tracks button. So let's say from here, you can click on custom tracks. And this will open up a pop-up window that allows you to submit your data. So what you will need to do is firstly give your data a name, so a track name. So we'll call this one example. Our species is human and our assembly is GRCH38. That's selected for us by default because we're in a location tab for the human GRCH38 genome assembly. And then we can either paste the data directly into the text box, upload a small file that's less than 20 megabytes, or we can put a URL directly into this text box if we want to upload a large remote file. We just want to copy and paste our data into the box. So you can see here, this is the bed file format. So we have these columns. The first column is the chromosome number. Column 2 is the start coordinate, and column 3 is the end coordinate. And column 4, we've just given a name to each of our regions. So P1, P2, and P3. The final thing that you have to do is select your data type, so your file format. So from the drop-down menu, you can see that obviously Ensembl is trying to work out what format we have here, so we need to choose bed. So that's actually everything we need to do, and then we can click Add Data. And we get a message telling us that the file has been uploaded successfully, and that we can either stay in the current region or we can go to the nearest region with data. So that's on chromosome 5. So we can click on the link in the pop-up window. And that will jump us to the location within the human chromosome 5. We can see the location has now changed in the location tab. And the page at the bottom, the region in detail view is just loading. Perfect. So what we can see now is that the page is loaded, and alongside all of the publicly available tracks, we can now see a new track here called Example. And yeah, we see a new track called Example, and we can see the discrete blocks that we described in our 3 rows of data. So we have P1, P2, and P3. Now, obviously, we can't see the full length of each of these features. So I'm just going to zoom out using the zooming function at the top right-hand corner here. And this will allow me to zoom out to view a bit more context around our regions of interest. Here we go. So now we can see that we have P2, P1, and P3. So now we have our genomic regions that we can see in a bit more detail. And we can see that they're all aligned to the genome as all of the other tracks are. And we can perhaps begin to see where these deletions map to these other features, such as the conserved regions, and importantly, the regions within the genes themselves, so the coding regions perhaps. Good. Okay. So that brings me to the end of the final demo. So just to wrap up, Ensembl is a genome browser which integrates the 4 main types of data that we've been thinking about. So the gene annotation, the variation, regulation data, and comparative genomics. So we've looked at each of these 4 different data types at various points throughout the course. And all this data is organized in a number of different ways, and you can retrieve it in different ways depending on your programming ability and the size of the data sets that you want to retrieve. So we've mainly been focusing upon the Ensembl browser site. So we've got the main website, www.ensembl.org, the Ensembl genomes pages for the non-vertebrate species, the dedicated site for the GRCH37 archive, and then all of the other archives as well. Then we've got the BioMart data mining tool that we just looked at a moment ago. And then you can also think about exporting the data programmatically using the Perl API or the REST API that Emily's going to go through in the course next week, if you know Perl or any other programming languages. You can also actually query our databases directly. If you know MySQL, you can query our databases directly using our MySQL servers. And finally, if you want to retrieve genome-wide data, you can retrieve what we call flat files from the FTP site. So if you follow the link here on the slide, it will take you to our FTP site where it's a public directory of files that are organized that have genome-wide data. So if you want to download the human genome sequence, for example, as a reference data set that you want to align data to, or you want to keep as a reference, for example, then you can download it from the FTP site. The only thing to notice is that the files on that FTP site are very, very large. They represent data across all of the whole genome for each species. So they're going to be very large and take a little while to download. So if you want further help in documentation, there's a number of places you can go. So firstly, we have a lot of online courses. So just to explain to you a little bit of background, Ensembl is part of the EBI, so the European Bioinformatics Institute. That's the institute that Ensembl is part of. So if we go to the EBI homepages, many of you may have been here before already. You can see that, yeah, so this is our institute's homepage. And on here, in the gray bar at the top, you can see that we have a lot of different services available. So if you click on services, you can find all of the different tools and data resources that we have ready for you to use free of charge. So we've got lots of tools, so like alignment tools like Cluster Omega, into ProScan and BLAST and Hammer. And then we have lots of different data resources. So we've obviously been focusing on Ensembl, which is interested in genes and genomes, but then we have lots of other databases focused on different types of biological data. So UniProt for protein sequences, PDB for protein structures, Europe PMC is for literature and citations. Expression Atlas we already looked at in the demo as well, for example, has information about tissue-specific expression patterns. So I would really advise you to spend some time and look through all the different resources that we have here to find the databases that you might find useful. If you go to training, in the bar at the top, you can find information about all of the training events through the EBI. So you can find them listed here. But at the top, if you click on Train Online, you can find all of the online courses that we have available through the EBI. So these courses sometimes cover a specific resource. So there are Ensembl courses that are here. Here we can see Interpro, for example. There's a course about UniProt further up here. So you can search for courses about specific resources, but there are also some more general courses with particular themes, such as variant calling or NGS data analysis, for example. So you can look at these courses and take them in your own time. So these are free to take. There's no time frame that you have to do them within, but there's no qualification that you get out of the other end. So it's just a really nice way to take responsibility for your own learning and find courses that are interesting to you to help you in your work. Good. The other thing that I want to point you towards, and I will do this in the follow-up email as well, is the Ensembl YouTube channel. So if you go to YouTube, and type Ensembl training, you can go to the Ensembl training YouTube channel and find videos, recorded demos, recorded webinars, tutorials of how to use Ensembl, access Ensembl data, and use the different tools as well. And finally, one of the most important things for me to mention is the Ensembl help desk. So if you do ever have any questions about using Ensembl, if you get stuck, if you're not sure about something that you're seeing in the browser, you can always email helpdesk@ensembl.org, and we'll always get back to you trying to talk you through your problem, help you with understanding what you're looking at, or resolving your query. So this is a really good resource to use when you're using Ensembl in the future. Then we have some public mailing lists, so dev@ensembl.org and announce@ensembl.org. These are mailing lists that are communities of people that are sort of helping each other out with specific developmental developer questions or announcements about releases and new data, for example. We also have a range of publications. So if you do use Ensembl in your work, we really appreciate it if you were to cite us. Obviously, Ensembl is free for you to use however you like, but if you cite us in your work, it helps us to know who's using Ensembl and what for. So it means that we can really drive the development of Ensembl in the future as well. So we have an annual paper in NAR. So this is the latest Ensembl 2021 paper. We have an Ensembl genomes paper, and then we have other papers about specific data types and tools as well that you can cite. If you've used the VEP, for example, you can cite the VEP paper. This is just to flag up to you as well that we obviously are running courses. At the moment, they're all virtual. This is an open course that anyone around the world could have attended. But if you want to organize a virtual course at your own institute, we'd be more than happy to organize that with you, that we can even run virtually as well. So you can get more information from the link at the bottom here, or you can email helpdesk as well to get more information. And then this is the final Ensembl slide with all of our team. So we're around about 100 people now in the Ensembl team. And I like to point out at this stage that all through the course I've been saying things like, we did this and we did that and we developed this tool and we developed that tool. But actually, it's I haven't done any of it. It's actually the hard work of all of these very clever people. So yeah, it's what we've been using throughout the course is what these people have been working to put together. Then we have obviously some social media channels. So we've got Facebook, we've got some Twitter channels, some Twitter accounts that you can follow as well. These are a bit more light-hearted with just some general announcements and information. But the YouTube, the Ensembl blog is really quite nice if you want to follow the blog ensembl.info. That provides information about any new releases, the new data, any job adverts, maintenance, downtime alerts, those sorts of things. So you can follow the Ensembl blog as well. And then finally, that brings me to the final slide. So this is a list of basically everyone that you'd have seen in the photo on the previous slide. And then this is all of our funders as well at the bottom here. So Ensembl is obviously a free resource. So it's actually down to the fact that we get funding from all of these organizations, which means that we can obviously keep developing Ensembl and making it available for everyone to use for free around the world. So yeah, without them as well, Ensembl wouldn't be possible as well.